I'm Shawn Kaeppler, Director of the Wisconsin Crop Innovation Center. The Wisconsin Crop Innovation Center is part of the University of Wisconsin-Madison. It's a crop biotechnology research facility where we work on using biotechnology to improve crop varieties. I'm here today to talk about hemp. We've developed a new method for hemp engineering and gene editing with the goal of producing improved varieties. So why hemp? Hemp is a crop that has had a long history. It was grown through the 1930s and then due to regulatory reasons couldn't be grown anymore, but there's interest in bringing hemp back into the production system. As we look towards diversifying cropping systems with all of the challenges that we're facing globally in terms of climate change and sustainability and food production, a crop like hemp is very important. Hemp is already a $6 billion global industry and it's growing quickly. Industrial hemp is defined as hemp varieties that have less than 0.3% THC. So hemp is not a drug. As the hemp market has grown, there's a need for new hemp varieties to meet the needs of the growers. Those varieties should be tightly controlled for a number of traits and for all of the purposes that hemp is used for. So we'll talk more about the different uses of hemp, but the control of those traits is very important. For instance, the 0.3% THC threshold is very important because if the crop surpasses that threshold, it has to be destroyed. Growing hemp is a balancing act where you have a number of considerations. Hemp is an unusual plant in that it has both male and female plants. The female plants produce seed and flowers and other types of things, and the male plants produce the pollen. But depending on the uses, you need the right balance of male and female plants. Hemp is also grown in fields, it's grown in controlled environments, grown in a number of different places. And when you grow hemp, you have to have the right balance of water and nutrients to meet your production goals. You need the right amount of fertility to have high productivity and high output of your product, but you also need the right levels of fertility to keep below the 0.3% THC threshold. There's also planting considerations and growing considerations. When hemp is planted close together, the hemp plants grow very tall and produce a large amount of fiber and a high proportion of stalks. When the hemp is spaced far apart, it produces a larger amount of flowers and more of some of the valuable chemicals such as CBD. In addition to growing hemp, you also have the harvesting considerations. If you're growing hemp for grain, you need a variety that will produce a large amount of grain and that will hold on to its grain so it won't shatter onto the ground before the combine can go through the field. Hemp for CBD, which is a popular medicinal compound right now, is usually primarily female. Only the female plants produce the flowers, and you want those flowers produced at a high density and to have a lot of CBD relative to THC in their oil. Hemp grown for fiber is taller hemp, usually produced for a large amount of biomass, and you want a high volume of fiber relative to other parts of the plant, high proportion of stalks relative to leaves and flowers, and you want that fiber to be of high quality. Hemp grown for grain, which can be used for animal feed or human food or oils, for instance, should have a high yield of grain. It should be primarily females, but you do need male plants within your hemp field to produce pollen to make those seeds. And you want the plants to hold on to the seeds and not shatter them onto the ground before you're ready to harvest. So in our hemp cultivars, we're interested in tightly controlling the traits such as cannabinoid production. Hemp for CBD should have a high CBD to THC ratio. CBD now has a value of about $2,000 per kilogram of oil, whereas other cannabinoids such as CBG has almost 25-fold higher value at $50,000 per kilogram. There's also an array of other cannabinoids that are found in hemp that also have medicinal value and have potential as we look towards the future of hemp for these medicinal purposes. We have developed a patent-pending method for hemp transformation and gene editing to develop new improved cultivars of hemp. The first step in the process is to create a gene construct. This is a circular piece of DNA that has our gene of interest that we're going to introduce into the plant, or the machinery to conduct the gene editing in the plant cells. We introduce that into explants, small pieces of hemp plants, isolated from hemp seeds, using agrobacterium. Those hemp cells with the newly introduced DNA are then grown into small plantlets is seen as hemp plantlet growing on tissue culture media. And then those plants are grown into adult plants where they can be clonally propagated or they can be grown to produce seed so that we can advance to the next generation and grow more of the plants that have the modification that we're interested in. This method was developed by a team of individuals working together at WCIC who have over 200 years of combined experience in plant transformation and cell biology. I am the director of WCIC. I'm a crop breeder and geneticist and biotechnologist. I work with collaborators on campus. Mike Peterson, who is a senior scientist now at WCIC focused on hemp biotechnology, and one of our point people for industry collaborations, and Shelby Ellison in the Department of Horticulture, who works on hemp genetics and breeding, has a vast amount of knowledge about hemp, and also conducts educational programs in the classroom and outside of the classroom related to hemp. We have substantial traction in this area. We've already received $200,000 of funding to develop and optimize the hemp transformation and editing method. We've produced proof of concept plants, including plants that express a red fluorescent protein and glow red when we put a special light on them with a special filter, indicating that they contain our new gene of interest. And we've also delivered products to industry partners and collaborators for genes that they are particularly interested in. In-house, we are working on plants that have altered and improved cannabinoid levels, including plants that have 0 THC, high levels of CBD, and high levels of CBG. This work in cannabinoids is done also with an industry partner. We're seeking new industry partners to look for other uses of hemp and to use our technology to make improved cultivars. We know that hemp can be used for fiber, for grain, for a number of purposes, and we believe that our technology can help to accelerate the process of producing cultivars for those purposes. We're also seeking partners who will commercialize the hemp varieties that we produce, because that's not a function that we normally conduct at the University of Wisconsin. WCIC has a substantial capacity as a facility with over 100,000 square feet total, 30,000 square feet of lab space, substantial, unique growth space for small plants and growing those plants all the way to adults and to produce seed. And so we have the capacity there to take on a number of projects, and we are seeking collaborators to help take advantage of that capacity and to advance us towards producing new hemp varieties. I'm Shawn Kaeppler. My team and I would be happy to meet with you and to identify collaborative projects with you. Thank you.